All right, everybody. This is going to be chapter 13, which is going to be meiosis. And you may remember in chapter 12, we talked a very small amount about meiosis, and we just said that it is going to be the division of gametes, which are sex cells, which for humans are sperm and eggs. Sperm in males, eggs in females. So first... one other thing we said is that instead of producing identical cells like mitosis, meiosis is going to produce different cells. And very specifically, we're going to have one parent cell that is actually going to end up producing 4 genetically different daughter cells. So it's the very basic outline of meiosis, and very importantly, it is a source of genetic variation, which is super important because that's how we get evolution, that's how we get new traits, that's how we get mutations. Like, this is how we can sort of, well, it's not how we get some mutations, but this is why we all look different, why every single individual is different. And the 3 methods, or the 3 reasons that it produces genetic variation is going to be the law of independent assortment, which we'll go into even more in chapter 14 with genetics, crossing over, which is a process that is going to happen in prophase 1, and then outcrossing, which means combining with different gametes, and that's, when a mom and a dad love each other very much, yada, 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 they each donate one gamete, so one egg from over here and one sperm from over here to produce a very unique cell that ends up being you. Wow, so cool. So those are the 3 ways that genetic variation happens. So gametes carry 23 chromosomes, and you may remember that somatic cells, which are the ones that we cover with mitosis, have 46 chromosomes. And you may remember in the mitosis video, I was saying that you get sets. You have a chromosome from your mom, and you have a chromosome from your dad for the same genes. And this is a homologous pair. That doesn't happen in meiosis. Meiosis, you're creating the one that you get from your mom and the one you get from your dad that eventually will combine, but that happens through sex. So that's not going to be part of meiosis. So gametes in humans carry 23 chromosomes. And again, so that means when these 2 gametes combine, when you get mom's gamete and you get dad's gamete, you end up with 46 chromosomes, and that's you. And it's also important that these chromosomes that you're inheriting become your homologous pairs. And so what I mean by that is this chromosome right here, dad's 3rd chromosome, which is this one right here, we'll say, and mom's 3rd chromosome, code for the same stuff. They code for the same traits. They might have different alleles, which we'll do in chapter 14, but you've already looked at chapter 14, so you know what an allele is. So that's sort of just the outline, and then just a quick review about chromosomes, chromatids, and homologous pairs. Remember that chromosomes is the number of whole structures that you see. So right here, I see a whole structure right here, and I see another whole structure right here. So that must be 2 chromosomes. And then over here, I left spaces specifically to write the numbers. Look at that. And then over here, I see 1 single structure and then this second single structure. So this is still 2 chromosomes. I also see 1 homologous pair in each chromosome, because this is mom's chromosome, this is dad's chromosome, this is mom's chromosome, it's just been replicated, and this is dad's chromosome, it's just been replicated. Or it's been through the S phase, I should say. Chromatids, though, over here I count 1 chromatid, 2 chromatids, 1 from mom, 1 from dad. But over here I count 1, 2 from mom and 1, 2 from dad for a total of 4 chromatids. Quick vocab review. Everybody loves a vocab review. So the reason meiosis sort of works is because it happens in 2 stages. So we have meiosis 1, and meiosis 1 is going to take a 2N or diploid cell, and let's write that, a diploid cell, and make it into a haploid cell. And that's meiosis 1. It's a reduction division, and that's going to be your homologous chromosomes are going to separate. So when homologous chromosomes separate, that's going to look like this. So let's say these are this is mom's chromosome, this is mom's chromosome, this is mom's chromosome, and then the rest are dad's. These are going to separate. So these ones will go this way, and these ones will go this way. And I've kind of demonstrated independent assortment here. You can see that not one cell is getting all of one chromosome. This one is getting, this gamete that we're going to produce on this side is going to have one of mom's chromosomes and 2 of dad's chromosomes. But this one over here is going to have 1 of dad's chromosomes, but 2 of mom's chromosomes. So you're going to end up with 2 cells. And we have 2 mom chromosomes over here, but only 1 mom chromosome over here. And so you can see we've gone from 6 total chromosomes to 3 in each cell. So this would be the diploid cell producing 2 haploid cells, or 2N to N. Now meiosis 2 is going to separate sister chromatids. So I'm going to take that one cell that had, we'll say it's this one here, I'm just going to bring it down, that has 2 of dad's chromosomes and 1 of mom's, and I'm going to split the chromatids apart. So I now have this. And I have a couple of moms and then dads. But remember, this is still the same amount of chromosomes in each cell. So there are 3 chromosomes in this cell. There are also 3 chromosomes here and 3 here. We've just split the chromatids. We've gone from this X and we've pulled it apart right down the middle and produced this. So we have 2 halves of the chromosome. All right, so we are not going to go super in-depth into all the stages of meiosis because they are the same as mitosis. So there's a prophase, a prometaphase, a metaphase, an anaphase, and telophase. They just happen twice. And there are a couple key things that we have to talk about that are going to change, though. And the most important stage that I want you to remember, no matter what, is prophase 1. Prophase 1 is where crossing over happens. So meiosis 1, when we go into meiosis 1, we have a 2N cell that has 46 chromosomes, but 92 chromatids. And we'll track that throughout. So in prophase 1, the normal stuff is going to happen. Chromatin is going to condense. You're going to start getting your spindle formation. Same stuff as prophase in mitosis. The really important thing, though, about prophase 1 is homologous chromosomes are going to pair up, meaning mom's chromosome here is going to like partner up with dad's chromosome. And you can see that they're actually touching at these 2 points. And those contact points are called chiasmata. And chiasmata, that is where crossing over occurs. And so what's going to happen here, and really all I need you to really know about crossing over, is that there's going to be a swap. So you can see that this chromatid right here is going to actually exchange some DNA at the chiasmata or at the synapse with this chromosome. So now we have a little bit of mom's chromosome here. We have a little bit of dad's chromosome here. So these are now different than the starting chromosomes. So that's prophase 1, crossing over. Remember it. It's super, super important. It creates recombination. I apologize, I'm still a little under the weather. So metaphase 1 is going to be homologous pairs are going to line up at the metaphasal plate. So you can see here we have the homologous pairs are lined up right down the middle, and they're going to split. And we're still 2n at this point. But very importantly about metaphase 1 is this is where independent assortment comes in again. Independent assortment. So independent assortment means the orientation of maternal and paternal chromosome for each homologous pair is random. And what I mean by that is if I have mom's chromosome 1, dad's chromosome 1, just because mom's is on this side doesn't mean the next chromosomes have to be that way. So the next chromosome might be dad's second chromosome here, but mom's second chromosome here. And when those split, then you can see how we already have some variation happening because we have a maternal chromosome 1 and a paternal chromosome 2. So they're not identical in any way. So this is now a new cell. We have not seen this configuration of chromosomes before. And then anaphase 1 is going to be the homologs separated. So those homologous pairs are going to be pulled apart, and then telophase and cytokinesis is going to happen and just split this cell and make 2 daughter cells that are now N in ploidy. So now we have 23 chromosomes in each cell and 46 chromatids. And remember, we started with 46 chromosomes. So we've halved the number of chromosomes. And then meiosis 2 is the exact same thing, except instead of these homologous pairs being pulled apart, we're going to actually pull apart these individual chromatids, just like in mitosis. So meiosis 2 is much more like mitosis. And then we don't need to really go through the phases because it's the same as mitosis. It's all the same steps, but I did include this just to sort of, I guess, point out the ploidy numbers. Should put technically 2n for anaphase because the cell hasn't split yet. But you'll see that we started with n, which is 23 chromosomes, and we ended with n ploidy, which is 23 chromosomes. And this was a short one, guys. This was a really short one. That is pretty much all I need you to know about meiosis for now. We will reference meiosis quite a bit in coming chapters. But for the test, I would hammer into your head these ploidy numbers. And I will be right back. I might have a little cheat sheet for meiosis. Let me check. I did not. Never mind. Sorry about that. But yeah, for the test, crossing over is probably the most important thing. Oh, never mind. I do have a cheat sheet. So this is the cheat sheet that I would say to sort of work on memorizing. I forgot to put key events, but I'll tell them out for you. So prophase 1, and this just tracks the ploidy number and the number of chromatids throughout all of the process. And there might be some questions about this on the test. So just kind of understand where the ploidy is at each stage. And then I really want you to remember crossing over. I want you to remember independent assortment in metaphase 1. I want you to remember homologous pairs separate in anaphase 1. And then in anaphase 2, I want you to remember sister chromatids separate. So this is sort of the meiosis cheat sheet that I think would be good to remember. And that should be it. Chapter 12 and 13 aren't super difficult. but they really lay the foundation for when I get into Mendelian genetics, gene linkage, all that good stuff. So I will see you in the next chapter.